Happy New Year, everybody, and welcome back to the Genetic and Genomics Lunch Seminar Series. Today, we are joined by Dr. Sarah Dyack and Christie Riddell. Dr. Riddell obtained her BSc in Biology and PhD in Biology from Queen's University in Kingston, Ontario, focusing on the molecular genetics of Drosophila. And then she moved to Winnipeg in 1982 to join John Hammerton's Somatic Cell Genetics Lab as a postdoc. And following that, became a research associate, offering to trade molecular skills for experience in human genetics research. In 1988, she joined the IWK Health Center, where she's been head of the Molecular Diagnostics Lab in the Department of Pathology and Laboratory of Medicine since then. And she's been overseeing the expansion of the lab significantly over that period of time. It started off as a two-person operation and was 10 people in 2012, and what is it now?
Well, we're probably about 13 or 14 now.
Keeps growing. And basically to meet the demands of the increasing demands on the molecular diagnostic services. Over her tenure, Dr. Riddell has been involved in several research projects, from characterizing tumor suppressor genes to local discovery genes via the ANGI and IGNITE projects. And she's also been active in research ethics, working on various committees over several years, most of which she has chaired at some point. So over to Sarah Dyack. Dr. Dyack obtained her BSc and MD degrees at Dalhousie University. She then did a residency in pediatrics at the University of Alberta, followed by a fellowship in clinical genetics at the Toronto Sick Kids. She's been at the IWK since 2000 and is currently the head of the Division of Medical Genetics. She's a consultant for the Inherited Heart Disease Clinic. She's a co-chair for the Maritime Newborn Screening Committee, and she's one of the co-investigators on the IGNITE project. So please join me in welcoming both Drs. Dyack and Dr. Riddell to talk about the perils of genetic testing.
So if I'm too loud, just let someone know, and I'll try to tone it down a little. I tend to talk a bit loud. So I'm Sarah Dyack. I'm a geneticist here in Halifax. Okay, and I'm really interested to have been given the opportunity to talk to you about some of the issues that have arisen over the last number of years regarding genetic testing, and I'm hoping to take you through the story of how this whole area has evolved, give you some good case examples, and hopefully improve your understanding of some of the things that we're facing in genetics in that regard. Okay. Now, this is a slide that one of my colleagues, Anthony Vandersteen, lent to me, and this is the long room. I think it's, if I'm not mistaken, it's the library at King's College in Cambridge or something to that effect. And so, I mean, this is to illustrate the fact that this is sort of like what our genome is like. It's this massive amount of information, almost too much to really comprehend, in many, large packages and in discrete packages as well. So it's like got multi-layers and huge, variable and extensive. And when we look at it in a little more sort of detail, we can, just make sure I can get this right, okay. So we can have big sections of the library. They're like the chromosomes that we can see under the microscope, and that's really where we first started to look at the actual molecular sort of, lab variation in genetics. We then developed techniques to let us look at sort of more sections of the bookshelves, and we would call those microdeletion syndromes, which we detected through FISH technology. And then more so, we have worked on actually reading the sentences that are within all these books and looking for errors in the recipe or the sentences that tell our body, of all the instructions to tell our bodies how to grow and develop and maintain ourselves. And we think of those as more of the information in the books. And this is sort of more the information that we get from DNA sequencing. And that's really what we're going to talk about today mostly, is the variation in the genome and how that makes it interesting to interpret DNA sequencing. So mostly this is going to be about single gene disorders. But there is a amount of variation in the genome that has become evident over the last number of years that we really weren't expecting. And when we first started thinking about variation in the genome, it was sort of like, we have a group here, and I don't know if you can sort of recognize this, but this is a group of teenagers that they all have Down syndrome. And that's caused by an extra chromosome 21 or trisomy 21. And so we thought, people could be somewhat variable amongst themselves with the same condition, but that ultimately the genetic basis of a lot of these things was going to be pretty straightforward. And with cytogenetics, just looking at the karyotypes for these individuals, most of the time it's pretty straightforward. They have the extra chromosome, they have trisomy 21 Down syndrome, and you move forward from that. Variation really wasn't a big issue. And as techniques improved over the years, there's been an improvement in the resolution in our ability to see differences in the copy numbers in our chromosomes. And so we started off with our classic karyotype, and then we got into the microdeletion syndromes. Probes were developed for us to be able to see microdeletions using these types of probes, where see there's 2 here and only 1 here. So we were able to determine there's a little piece missing there. And that cannot be seen by looking at the karyotype, because the resolution of the karyotype just wasn't fine enough to be able to detect those things. So we started developing these techniques to be able to improve the resolution and diagnostic ability in genetics. And this is one of the first examples, for example, in cytogenetics. And then as time has gone on, we now have incorporated chromosome microarrays into the mix, where that's even more of a fine resolution than the FISH testing. But with that increase in resolution, we have now realized that there is a much more of a variation in normal. So in chromosome microarrays, we are much more likely to see small deletions and duplications that are just common in the general population. And that was a big surprise in some regards to geneticists, because we really thought that it really mattered that you always had to have the same amount of DNA, you always had to have all those pieces there to be normal. And then we realized there's more variation in that regard than we thought. And this hence the issue of variants of unknown significance. When you find something and you honestly really don't know if it means something or means nothing. And so we're going to apply this to the sequence of the genome, not the copy number. So we're going to put aside chromosomes and copy number and just talk about what's the actual spelling, what's the code of the genome, and then how does that variation create so-called perils in the interpretation of our genetic test results. And this is all based on true cases and clinical experience. So it was quite a surprise when we realized that there was more variation even in the sequence than we had expected. So when I first started out in genetics, and it's a long time, but it's not that long really. I mean, I don't know, 15 years almost at the IWK. We were really not expecting that there would be a lot of protein coding differences in genes and that would be normal. So because of exome sequencing and the development of being able to sequence so many genes, we have now realized that the genome is not sort of a steady state, obvious code. It's highly variable amongst normal people, to the point that, and sorry, some of these things just got cut off in a couple slides, so hopefully it won't be a big problem. But for example, an individual's genome harbors at least 80 nonsense variants, and Dr. Riddell is going to get a little bit more about what that means and how that's important in how we interpret our genomes and in just regular gene testing and sequencing. We have significant numbers of severe frameshift variants, lots of splice mutants that can cause serious problems with gene expression. And on top of that, thousands of these missense variants, little spelling errors that are so common, yet some of them are very severe and are significantly pathogenic. And being able to tell what's just normal and what's disease-causing has created a huge challenge for us in genetics and genomics. And it has become quite challenging in certain circumstances to interpret the variants that we're finding in our sequencing. So, the objectives of this talk, I'm really hoping that by the time you leave today, you're going to have a little more understanding about how the molecular lab, or the DNA lab, assesses DNA sequence variants. And we're hoping to illustrate the utility and perils of the genetic testing and just give you some hints on what to be careful about when you may have a genetic sequence and you're trying to interpret it. And also to really hammer home the point that the interpretation of genetic test results isn't like a blood test. It's not like a CBC where you have a really good idea about how likely is this to be true, and you can just repeat the test and see what happens, and it's pretty straightforward. It's really important to interpret genetic test results in the context of the bigger picture of the patient, what the change is, and even in the family structure. So I really hope that that becomes quite clear to everyone. Okay, and now I'd really like to invite Dr. Riddell to come up, and who's a molecular geneticist, and tell us about that aspect of the situation.
I need one of them. Okay.
So to ease in the transition, I've been asked to use this microphone, so I'm probably going to put it down and forget it somewhere. I'll try to keep it with me. So what I'm going to talk about is probably going to be a review for a lot of you, but I just want to make sure everybody's at the same starting point before Sarah dives into the clinical examples and tries to draw correlations between what I detect in the molecular lab and how she takes that back to her patient and family. So before I actually start, I want to say that the theme through this presentation really relates to what we might call the low-hanging fruit of Mendelian genetics. These are single gene, large effect. For the most part, we're going to talk about autosomal dominant, but they can be autosomal recessive, well-characterized gene/disorders. So we won't actually say that many times, but that's pretty much what we'll be referring to. So when we get this information, we get it in a sequence, obviously, and we compare that sequence to the known sequence, the reference sequence that's in the databases, NCBI or Ensemble or UCSC. And we see a lot of variation between what we get and what is the reference sequence. And the question is, the variation, the differences, are they functionally significant or are they benign variation? So we call the variants, we call them sequence variants, and we categorize them either into polymorphisms, which is to say some relatively benign, maybe not totally non-functional, but relatively benign change that is present in the population, usually at a relatively high frequency, and a relatively high frequency means anything over 0.5%, that's a soft number, that 0.5%. Mutations, on the other hand, are variants that have a pathological functional change. Usually they're very rare. However, in the context of common genetic disease, they may be present in the population at 0.5 or 1 or even 2%, depending on the condition that you're looking at. So that cutoff between mutation and polymorphism in terms of frequency is a very soft boundary. So mutations that cause pathology, which are the ones that we want to be able to identify. We can identify the change, but can we identify whether it's pathological or not? Do they change? Does this sequence change the final coding sequence? I think you can follow it. There's too many buttons on this. I have to figure out which one it is. It's the middle one?
Yeah.
The middle one?
There you go.
There we go. Oh, I see. It's a slow response. Okay. So change in the final coding sequence. And there's a variety of changes that we can categorize. They can be a nonsense mutation or frame shift. Sarah's already alluded to these. Nonsense is an amino acid change to a termination codon and causes a premature truncation of the protein. And obviously that means that from that point on there is no protein synthesized. So if that change is early in the translation, then there's virtually none of that product generated. Frameshift mutations are mutations that don't actually cause a termination at the site of the mutation, but generate a frameshift in the reading of the codons downstream until eventually a termination codon is found, and then the termination occurs. So it goes from normal sequence to garbage to termination, and that usually gets degraded through the nonsense-mediated RNA degradation pathway. So those mutations commonly are what we might call null mutations. That is to say, there is no transcript, there is no protein product generated from those. Splice site mutations are another form of mutation that can either generate a protein which is missing a piece or has an extra piece in it, and may or may not downstream generate a frameshift, because it doesn't necessarily maintain the reading frame of the protein. And then missense mutations, which Sarah talked about, which are what we might call typos, where you have one amino acid changed for another. And depending on what the change is and where it is in the protein, they may or may not have very significant pathological phenotype associated with them. So those are, whoops, this is too slow for me. So those are discussions of the changes in the coding sequence. You can have the same normal coding sequence and change the quantity. So normal structure, abnormal amount, and those could be regulatory mutations, they could be deletions and duplications of entire genes, or chromosomal regions. They could be the result of imprinting or epigenetic effects where the sequence is normal, but the superimposed regulatory mechanism on top of it does not allow normal expression. And you could have mosaicism, where you have different expression. You have different gene structure resulting in expression at different levels, and this may be a quantity issue, it may be a tissue-specific issue. So again, adding a layer of variation onto the actual expression of that gene. In addition to these relatively well-characterized changes, there are emerging novel pathogenic mechanisms. In terms of regulatory changes, antisense RNA is becoming better and better described as not only a biological mechanism of regulation, but an excellent therapeutic intervention vehicle. In addition, there's microRNAs, this is more emergent, and long interspersed non-coding, LINC RNAs. The other thing I want to mention, from my point of view, as somebody who's been involved in the field for quite a while, we always decided that if the change was synonymous, that is to say it did not change the amino acid translation, then it was unlikely to be pathogenic. We could not envisage some way whereby it could be pathogenic. We now know that there are synonymous coding changes which actually affects splicing, maybe introducing additional splice site donor or eliminating splice sites, and therefore, in fact, now a few examples are pretty well characterized of synonymous changes having pathological effects. So, as I use the term emerging, there are probably going to be other mechanisms that we don't know of at this point. And why is this important for interpretation? Well, if we're talking about a variant that has not been previously described, how are we going to move towards deciding whether it's pathological or not? Does it have an extreme phenotypic, does it have an extreme molecular change? Does it disrupt the protein structure? Are we looking at a novel mutation in a gene that's well characterized? Are we in a gene discovery project and we're trying to figure out what a predicted functional change is in a predicted protein function? So these are layers of variation that we have to contemplate. So we know what the variant is, so now how do we look at it? Where in the gene is it? Is it in a functionally important site, catalytic or active? Is it part of an essential structure? Does it help maintain the tertiary structure of the protein? Does the protein interact with other proteins, as in a multimer, and by changing an amino acid, you're changing an association of this polypeptide with that polypeptide? Does it cause changes in splice sites? Because that can result in a frameshift mutation, which can change the overall protein structure, or even result in a null protein, a null phenotype. How do we evaluate the missense changes? Amino acid size, polarity, acidic, basic. There's a whole variety of means by which we can compare them. Is this amino acid in an evolutionarily conserved region of that protein when you look at the family that this protein belongs to? Obviously, we've already mentioned synonymous versus non-synonymous. In the protein, in this in silico tools that we use, we're really, for the most part, looking at translation. So we're looking at the amino acids that are changed. There are no really good software tools to look at how synonymous changes affect splicing at this point. So I'm not going to actually mention those. But so we have tools that do all this comparison for us. And these in silico tools, in silico prediction tools, are useful, but they are guidelines. So the most common ones we use are SIFT and polyphen. So SIFT is sorting intolerated from tolerated changes. Very simple, straightforward database. Polyphen compares these various components of functionality of amino acids within a protein. And then they give you a likelihood of this change you're looking at being pathogenic or non-pathogenic. The ASSA is a splice site prediction program, which is useful and gives you an idea of whether the variant that you're looking at in the intronic regions is may in fact affect splicing. Very useful. We like to have numbers. If we see something that's 99% likely to be pathogenic, we say, yes, that's great. We must keep in mind that these, even when they are all in agreement, they are not 100% sensitive or specific. And so they really simply point you in a certain direction. And the most important things really are to remember certain essential considerations. How common, if you see a variant, is it common in the general population? Maybe that's an indication that it's not pathogenic. Doesn't matter what everything else says. Do you have functional data on it? Does it disrupt the function of the protein? And never forget that this is genetics. Does it segregate with the phenotype? And Sarah's going to take that and run with it because the phenotype may or may not be 100% penetrant, et cetera, et cetera. The take-home message from my little part here is that the determination of pathogenicity is a moving target, and as our knowledge base increases, so does our definition of how we determine pathogenicity. So, back to you, Sarah.
Thank you, Christie. And so, for me, a genetics talk is just never complete until you can put up a few pictures of kids with rare syndromes. So I just love these syndromes. But this is an example of where I'm going to take you through sort of where genetics has been very successful in helping us with diagnosis, management, prenatal diagnosis, etc., and then move you down the line to where it's becoming a little more problematic. So we're going to start off where genetic testing has been famously successful. And, rare syndromes with a recognizable phenotype, genetics usually is finding, when we get variants in these genes many, many times, the majority of them are going to be pathogenic. And this is an example of a child with Kabuki syndrome, Noonan syndrome, Waardenburg syndrome, Camille-Delange, and a deletion 22, and that's a chromosome one, but the point is that the syndrome rare, it works. And we've been very successful in that regard. And other non-syndromic single gene disorders, we have had a great deal of success with genetic testing and issues with variants. And common things like cystic fibrosis, an autosomal recessive condition where children are affected with lung and digestive problems and it shortens their life. Duchenne muscular dystrophy, a condition where this happens to be X-linked, so boys are more affected, women can be carriers, very severe disorder. Children lose the ability to walk, wheelchair bound, and die in their early 20s. So really difficult, and genetic testing has been very helpful in the treatment and management of that condition, along with other conditions such as spinal muscular atrophy, myotonic dystrophy, and some of the more adult cancer syndromes, been very helpful. And it kind of, these are the first conditions that in genetics we were really using genetic testing commonly in. Because these are the ones where they found the genes first and we were able to implement them. And at the time, you really kind of got the impression in the earlier days that, you know, when you found something different in these genes, that it was like finding the golden egg in like this sea of the same. Like it seemed so obvious, like how could you miss that? It must mean something. And we got lucky because most of the time it did mean something. And I think it's because they were rare, easily detectable phenotypes with other tests that we could prove that we were on the right track. And it worked out rather well. But it didn't always work out well. And here's an example of where in the earlier days, in around 2001, where things went a little bit wrong. And just to give you sort of just a little hesitation, just so that you know nothing has been perfect here. There was a mutation in the cystic fibrosis gene, CFTR, that was reported called I148T. But as time went on, and this was done in more and more people, in more affected people, and in more people in the general population, it became obvious that this wasn't a mutation that was supported by clinical evidence. But before that became the case, it was recommended in a mutation panel for cystic fibrosis genetic testing and screening that was implemented in prenatal diagnosis, being planned for doing pre-conception counseling for families. So this was a kind of a big deal that this was not accurate. And ultimately what was discovered when people with this mutation were found to have a severe mutation but didn't have cystic fibrosis, that there was another mutation that was commonly found along with that more common polymorphism. And that was a more severe mutation. That was actually the severe mutation that caused cystic fibrosis. So they had to, through various laborious efforts, went through all this information and then took this mutation off the recommended cystic fibrosis panel. So this is to give you an illustration of the point that even with the best evidence we have, and with national bodies making these decisions, there is the chance that some of these things could be in error, because there may be an alternate explanation, such as another mutation, which might have been the explanation for the problem. And in those days, we really didn't have a lot of good evidence about what was common and what wasn't common. So you can see how that could happen. And interestingly, this same mutation was implicated in the development of hereditary pancreatitis. And then once they realized this, they did more frequency studies looking at this in the general population, and this is the same frequency as in control populations. So there's really not even a real correlation. So it was a sort of a selection bias circumstance. We test for CF, we find this change. Well, because there are two rare things, it must mean that it's the cause, but that's just not always the case, and we just have to keep our wits about us. The next big area where people can go down the wrong path is in the interpretation of negative test results. And I really like this cartoon, so I'm going to read it out to you, because it does really sound like what we say in our clinic. It's like this, Mrs. Cameron. The results are negative, but that doesn't mean not positive exactly. Nor is it not negative. We wouldn't want a double negative there, would we? So it really does feel like that sometimes in genetics. I know the counselors are kind of laughing because it is sort of like that in clinic. Negative doesn't necessarily mean there's no mutation, that you're really negative. You need to have this in the context of the bigger picture to be able to interpret that result. So here's an example of a negative that wasn't really a negative, okay? So we have, this is for Duchenne muscular dystrophy, that neuromuscular disorder that's really severe. It's X-linked. Boys are more severely affected, women can be carriers. This lady had a son with her first husband who unfortunately was affected with Duchenne muscular dystrophy. And at the time in the early 1990s, what have you, when that child was born and then developed Duchenne muscular dystrophy, genetic testing was performed. But at the time, the genetic testing could only pick up 2/3 of the mutations in the DMD gene. So this child had a negative genetic test. And unfortunately, that translated into that, for various other reasons, that the mom, they didn't feel the mom was a carrier. 20 years later, she has a new partner, another son. child affected with DMD. We repeated the genetic testing, and a causative mutation was found in all of these 3 individuals. And I guess the point that I want to make here is that you have to be careful when you interpret negative test results and then try to figure out who else in the family is at risk. You have to be really careful about the fact that there could be things that you didn't detect, even though you got a negative result. And also that the genetic testing capability is evolving. And so if you've had a family that had a negative test, you might want to check and see if there's new testing available, because new testing became available here, and then we were able to sort this out for this and the extended family regarding carrier status for this. So negative doesn't always mean it's negative, and that there could be alternate explanations, and that also check up on what's going on in genetics, because there may be new tests available that weren't at the time that you originally were investigating the family. Okay, so those are kind of the more straightforward ones with a few, caveats to be careful about. Now we're going to go into genetic testing in dilated cardiomyopathy. And I'm picking dilated cardiomyopathy because I do see a lot of individuals with inherited cardiomyopathies through my work with Dr. Martin Gardner and Chris Gray through the Inherited Heart Disease Clinic at the CBHA. We have a collaborative clinic, and with some of the genetic counselors, Amy and Natasha. So this is a condition where there's more of a vague phenotype. So you can have a dilated heart, but the question is, well, what's the cause? It's not like Duchenne muscular dystrophy where that's the only gene, it's clearly you can make a diagnosis. In dilated cardiomyopathy, it's a heterogeneous clinical disease, so there's many different things that could cause it. One of them is genetic, other things such as heart disease, alcoholism, for example. So there's lots of things that could cause dilated cardiomyopathy that aren't necessarily genetic. And then it also shows an enormous amount of what we would call genetic heterogeneity or variation, in that there is like more than 50 genes, individual different genes, and I'm not talking mutations within one gene, I mean 50 separate genes that have been implicated in the development of dilated cardiomyopathy. So when we go from something that's more rare, more thoroughly investigated, more researched, the phenotype is easy to tell, proving what it is by non-genetic means on a lot of circumstances. And you go to a condition where you're wondering, okay, is this genetic, and can we use genetics to help this family? When we go to something like DCM, which has this incredible heterogeneity, we can run into more troubles when it comes to the genetics. So I'm going to start off with one of my success stories. And this is actually a family. It's the first consult I ever had as a geneticist here at the IWK was this family. And my first consult ever as a staff, which I'll think back, it's a little intimidating, was this guy here. And he showed up when he was 13 with dilated cardiomyopathy in the ICU at the IWK. And I did a lot of work tracking down all the family records. They had been followed in the IWK and in genetics, some of these people, over numerous years. And I was able to figure out that it was likely that this family had a mutation in the LMNA gene, and I'm just going to call it lamin, just because it's easier for me to say. It produces a protein called lamin AC, so I'll just call it lamin, but so that you know that that's the situation. And we sent that off, and in those days there was hardly any gene test you could send off. This had just recently been described, so I think I probably sent it off to a research lab. I don't know if it was clinical or research. It might have been just barely clinical at that point. And we found a variant in this gene, and it was a missense variant. So now thinking back, I'm like sort of horrified. It really could have gone wrong. But the beauty of this was I went over to PEI and I examined like the entire family. And we did genetic testing in everyone. And fortunately for me, the mutation followed perfectly, segregated perfectly with the phenotype in the family. And in fact, we predicted numerous individuals in this family that they would develop dilated cardiomyopathy. And we've had a good success in that regard. So I'm pretty confident because of the segregation data that this is correct. It has not been wrong. Interestingly, I've collaborated with someone in Ottawa, and they did some functional analysis on this family, and the functional data that they were looking at did not support pathogenicity, as it turns out. And my other caveat is that one single piece of data, functional or, looking at the SIFT and polyphen, each of those things in and of themselves doesn't mean anything. You have to look at it in the whole context of the situation and come up with your best, opinion in that regard. But segregation is really a strong one. So this worked out great. We were able to prevent a young lady, this woman here with 3 young children, from having a fatal arrhythmia. I mean, it was wonderful. So I felt really good about this. But as time went on, things got a little muddier. More genes became available. Things turned in from testing like maybe one gene or a single gene to these panels. We learned more and more about the different genes causing dilated cardiomyopathy, and now we can test many, many, many genes all at once, looking for things that cause this in the family. So here's an example where it got a little more confusing. So this is a mother and father had these 2 boys, and both of them developed a significant early onset dilated cardiomyopathy. And so this, unfortunately, this child had already passed away directly as a result of this dilated cardiomyopathy, but we were able to see this individual. And so we sent off testing for dilated cardiomyopathy, and I'm going to call it DCM from now on if I can remember. And so we got this test back. It was LMNA, and I thought, great, I have experience with lamin, so, I was feeling confident. The variant was a missense mutation, but it was literally noted in the report to be presumed pathogenic, and that was from 2009. So honestly, that's really not that long ago. So this was an experienced lab. So what we did was we then tested the parents, and it was the father who carried the mutation. But looking at the family history, that was a problem for me, because the father has 11 siblings and numerous, huge amounts of family members, and not one of them had dilated cardiomyopathy. Father himself was not affected, and there's a hugely strong family history of DCM on the other side of the family. Mom herself isn't actually diagnosed with DCM, but she has some serious heart problems and heart failure. So that's a bit suspicious. So I wasn't really convinced that this lamin mutation at this point, even though it was strongly suggested in the report that it was the cause, I didn't believe it, because this doesn't make sense to me. The segregation doesn't work. And so you can imagine that if you don't have large families with this type of helpful information, that the interpretation of that would have been much more difficult. What if they had a small family? It would have been very difficult to figure that out. We told the lab about that, and we updated the report that this was of unknown net clinical significance now related to our information that we provided back. There was updated testing that became available in December 2012. We sent off a pan-cardiomyopathy panel with 51 genes, and there's even more now. They found a totally different mutation in something called Titan, which they thought was likely pathogenic, giving us an alternate genetic explanation for the DCM in the family, and it's segregated in 2 affected family members. The lab changed their report to say that the lamin mutation, that the available evidence supports that it is most likely benign. So even though the report says pathogenic, sometimes it's not always pathogenic. And what about this? Is this non-penetrance or not pathogenic? A lot of the times with dominant conditions where you only need one of the mutations in a pair of genes to develop the disease, there's this thing where you can have that change, but you never develop the disease. But you pass on that change, your children are at risk to develop the disease. So that's non-penetrance. That makes the interpretation of all these variants much more complicated. So we have this gene, ABCC9, that was added to this pan-cardiomyopathy panel. So everyone with DCM that goes to that lab gets tested for this gene. The reason they added it is because it's supported by functional data and in a Nature Genetics paper. But interestingly, they did not do segregation for the mutations that they found in their 2 small families that they published. So we got the report back, presumed pathogenic. It's a stop codon, but very early on in the gene, so indicating a totally null mutation most likely, which wasn't necessarily consistent with what they found in that paper in those 2 families. So dilated cardiomyopathy, We tested as many people as we could, and 1, 2, 3, 4, 5 individuals in this generation at the ages of between 30 and 60 have not developed any hint of dilated cardiomyopathy, even though they have that mutation. Unfortunately, I was a little worried about this, so I suggested the whole family get tested and screened clinically. But none of the other individuals in the family who got the testing that was negative bothered to go get their cardiac screening, although that was recommended. And so I've been burned a few times that I don't necessarily think at this point, I'm not sure if this is non-penetrance or non-pathogenic, but I'm pretty concerned that this is not pathogenic and that we don't really know what the reason for that DCM is in that family. So that creates a whole host of problems. So, pathogenic mutations, the standards have evolved over time. What we used to know and think has changed. As the time goes by, we have to reassess how we interpret data that's new and even from families that we are seeing in follow-up from the past. Severe mutation, functional data, and a high-impact publication does not ensure that that mutation that was predicted is pathogenic. And the family studies of segregation are crucial. The other thing I learned was make sure the family members get their clinical assessment first so that people don't read too much into these variants where we're not 100% sure what they mean. And to kick it all off, there's some recent data. The NHLBI Go Exome Sequencing Project, or ESP, there's been some exome sequencing was done, and they gathered up around 6,500 people that had their exomes done, and they put it into a database. Now, those were collected from healthy controls, some people with extreme specific traits, like specifically LDL elevation or high blood pressure, or specific diseases like early onset MI and early onset stroke. So you have to just take that into consideration. But what they've done now is they're looking at these large population bases to determine how common are these pathogenic variants in this, quote, control population or in the 6,500 people. And before we used to think, well, if it wasn't found in 200 chromosomes, meaning 100 people, then we felt, okay, well, it's rare, so it must be pathogenic. Well, looking at this, for example, hypertrophic cardiomyopathy, dilated cardiomyopathy, and RBC are all sort of cardiomyopathy type of things I see in clinic. And when they looked at all the reported pathogenic mutations in the literature, okay, for these three conditions, 14% of them were found in ESP. 18% of them for these two were found in the ESP. Okay, if you look at carrier rates, there's like 1 to 6 to 1 in 4 people in these control groups have a quote-unquote reported pathogenic mutation. So I think what this illustrates is that there's a lot of misinformation in the literature. There's a lot of mutations that are reported as being pathogenic that aren't, because we used to think that something rare, less than 1 in 100 or 1 in 200, meant that it was disease-causing, and that's not the case. So it's really, really important to understand that and reinterpret, look at the literature and make sure that whatever that they base their assessments on as pathogenic still breeds true, okay, compared to what the original lab. So say you see someone in follow-up and it's 5 years later. This is a problem for you. So, some people talk about using population screening, using exomes to find people who are healthy and so they can get them, what's your risk? I would approach that with extreme caution. We would have no way of how to interpret these variants. I can barely interpret them when I have families with 11 siblings. So personally, I don't think right now it's the way to go until we get some more understanding of natural variation. So I'm just going to end up with, as you can see from your genetic printout, you only think you're depressed, whereas you are in fact a jolly, happy, full of the joys of spring type person. So we can't always trust what the genetics says when it comes to basic common variants. We need to understand normal variation and be a little skeptical if it doesn't follow what we're looking and seeing at us in the family and in the context of the situation. So in conclusion, don't get me wrong, genetic testing is excellent. It has been superb in the genetic diagnosis of many, many, many families, almost all of them. But there are some caveats to that. the determination of the pathogenicity needs to become more stringent, and is becoming so as we understand stuff about how normal variation really works. So most of our genetic tests are superb, but you have to think about pathogenicity and why, because some of them aren't as good as others. And don't let the genetics take you down the wrong road. So my husband told me, okay, when the Russians came in after World War II to the Czech Republic, that the Czechs turned around all the road signs so that they could confuse the Russians when they were invading their country. So don't let genetics confuse you. Don't follow the wrong sign, okay? You have to look at the map. You have to look at the bigger picture too. And genetic tests are really a puzzle with many pieces that you need to put together to come up with your conclusion. So if there's anything you take away from this today, please just look at genetic tests and interpret them in the context of all these other things we've been talking about. So, thanks very much. I really appreciate the opportunity to speak to you today. And thank you to Dr. Vandersteen for the some of the slides, and also to Andrea Rideout, one of our genetic counselors, for helping with some of the pedigree PowerPoint slides. So thanks again. Questions? Comments?
Hi, it's a great talk. Just a general questions. What is more important, whether or not this is a rare gene versus a rare disease? And I presume a rare disease is more important.
Yeah, so maybe I didn't make that clear. So so for rare diseases, most of the, so there's a couple things. If it's a rare disease with basically a very minimal amount of genetic heterogeneity, like it's one disease and you know it's caused by one gene, it makes it a little more easy to interpret the data. If you have a rare disease that's an obvious phenotype, and you're pretty confident whether they have the condition or not, it makes it a lot easier to interpret the data. But if you have something that's pretty vague, like say epilepsy, so you have a child come in and they have this rare form of epilepsy, or it's like a seizure disorder or what have you. There's so many different genes that can be involved in the development of epilepsy. It makes it much more difficult to tell what's normal variation sometimes versus a pathogenic change. And so we just have to take that context plus what the mutations say and put it all together and give our best shot to it. And then be cautious if we're going to use that for prenatal diagnosis if we're not sure. So the last thing we want to do is use these tests in a way that isn't accurate for those types of major decisions. So I don't know if that helped to answer your question. Christie, do you have anything else to add to that?
Just curious.
I mean, we just kind of got lucky with the rare ones.
So if one is more important, then what's the purpose of doing genetic testing in a healthy, normal population?
I would say at this point, I wouldn't in a healthy, normal population, because then you can't interpret the results. It makes it very difficult. Now, if you have something like breast cancer, we often test healthy people for breast cancer. But the difference there is that we start off with someone who's affected with the disorder. So if you have someone who's affected with the disorder and they have a clear mutation that's pathogenic, then you can test other family members in the family and tell them whether they're at high risk to develop it or not. But you can't test the healthy person first and then always be able to interpret those results. That's where you end up with a lot of problems. Yeah. So I'm pretty hesitant about just doing regular. The ACMG is interested in having labs report pathogenic mutations for a lot of these cardiac problems. And personally, I'm a little concerned about that because I'm not sure how everyone's going to be able to interpret those results. And then how do you tell if it's a problem or not? There's insurance implications. There's turning someone who's healthy into someone who's quote-unquote ill. So there's a lot of problems with doing this wrong.
I have a
question. One area that I didn't hear about is how do you handle specific subpopulations, like let's say natives.
If you have a missense mutation, then you're trying to interpret what it means, and you're looking at a population that's quite specific in terms of the frequency of a missense mutation, and you're comparing that to the general population.
Yeah, so I mean, at this point, I would just, the risk of having an isolated population is that there could be rare variants that are just common in that population that might not have been seen in Northern European databases, for example. And so the risk there is that by finding something rare, you might misinterpret it as something that's pathogenic based on the frequency of it in a different population. So that's why the ESP data is really important, because they have over 2,000 African Americans in the sample. And so you're getting better data for the African American samples, which could potentially, like if you didn't have that group and have a large population there, you could have a higher chance of misinterpreting the results. So for someone like the natives here, where we're not going to ever get that, we just have to keep that in mind in the context of the situation, and then think of that when we're interpreting the results.
So thank you.